Hello everyone, welcome back. Today I am here with one important topic of plant embryology, that is embryo sac or female gametophyte. In this presentation I will try to cover how the development of this embryo sac takes place, and on the basis of some variation, this development of embryo sac is of three types. One is monosporic embryo sac, then bisporic embryo sac, and tetrasporic embryo sac, that I will discuss one by one in detail. Then I will discuss the structure of mature embryo sac, and finally its various component. So these are the topics that I will try to cover in this presentation. Let's start. Firstly, this embryo sac is an oval sac-like thin-walled structure which is present towards micropylar end of the plant ovule. So ये जो structure है embryo sac, ये एक oval sac-like structure होता है जो कि thin-walled structure है, and it is present towards the micropylar end of the plant ovule, and it develops from functional haploid megaspore. और इसकी जो development है वो होती है from the haploid megaspore, which is the first cell of gametophytic generation, और एक ही functional megaspore से बनता है ये. Embryo sac contains 8 nuclei in most of the angiosperm. जो commonly angiosperm में embryo sac होता है उसमें होते हैं 8 nuclei and 7 celled. So mostly 7 celled and 8 nucleated embryo sac is found in case of angiosperms. So there are 3 cells which are present on the micropylar end, और इसे कहते हैं egg apparatus, जिसमें से एक होता है egg और 2 होते हैं synergids, while 3 are on opposite pole towards chalazal end, जिन्हें कहते हैं antipodals, and in the center it contains one cell which is known as central cell. It contains 2 nuclei. So total 8 nuclei, 7 celled structure is present in most of the angiosperm. Then egg apparatus is towards the micropylar end, and antipodal nuclei occurs at the chalazal end. So जो antipodal cells हैं वो होते हैं chalazal end में. Then 2 nuclei are at the center region known as polar nuclei. इन्हें polar nuclei कहते हैं. They also form the secondary nucleus. इसे secondary nucleus भी कहते हैं. Then egg nucleus and polar nuclei develops into embryo and endosperm respectively. जो egg है after fertilization it will give rise to, जब egg fertilize हो जाएगा with one male gamete, then it will give rise to zygote, और जो endosperm nuclei है they will fuse with the second male gamete and they will give rise to triploid endosperm, and it helps in nourishment of developing embryo, and this process is known as double fertilization. इस प्रोसेस को बोलते हैं डबल fertilization. Next comes development of embryo sac. As we know that megaspore represents the first cell of female gametophytic generation. जो female gametophytic generation है megaspore represent करता है उसका first cell, and this functional megaspore lies toward the chalazal end. जो chalazal end है वहाँ का linear जो tetrad है megaspore का, वो जो chalazal end वाला megaspore है that becomes functional and give rise to this female gametophyte. But there are some exceptions. कुछ exceptions है like family Oenothera. There is family Oenothera जिसमें जो micropylar end का megaspore है that remains functional. Oenothera के members में जो micropylar end वाला megaspore है that remains functional, while in case of rosa, जो rosa है rose, उसमें second cell of this tetrad becomes functional and give rise to this female gametophyte. And the details of the embryo sac development was first studied by Strasburger in 1879 in Polygonum. So Strasburger ने सबसे पहले स्टडी किया है embryo sac की developmental details को in 1879 in Polygonum. इस प्लांट में इन्होंने सबसे पहले स्टडी किया है. Then the functional megaspore enlarges in size and divides by 3 mitotic division. जो megaspore है functional megaspore out of 4 only 1 remains functional और बाकी 3 disintegrate हो जाते हैं. So this functional megaspore enlarges in size and divided in by 3 mitotic division resulted in 8 nucleate structure. Firstly, the first mitotic division. After first mitotic division, जो 2 न्यूक्लिया है they moves towards the opposite pole. वो दोनों ही opposite pole की तरफ move करते हैं और opposite corners की तरफ of the embryo sac. A central vacuole starts appearing. एक center में एक vacuole appear होता है. And after 2 mitotic division, जब 2 mitotic division हो जाते हैं then it results in formation of 8 nuclei. After 2 mitotic division, जो फर्स्ट डिवीजन है दैट इज रिपीटेड बाय मोर 2 mitotic division जिसके रिजल्ट से बनते हैं 8 nuclei. So these 8 nuclei are arranged in such a way that they are organized into 3 groups, that is 3 plus 3 plus 2 on micropylar end, chalazal end, and 3 in the center respectively. So 3 are at the micropylar end, 3 in the chalazal end, and 2 in the central cell. So 3 nuclei are at micropylar and constitute the egg apparatus. जो micropylar end वाले 3 सेल वो बनाते हैं egg apparatus which consists of egg and 2 synergids. While at chalazal end there are antipodals, उन्हें कहते हैं antipodals. And this structure becomes 8 nucleate 7 cell structure which is known as embryo sac. और इस structure को कहते हैं embryo sac. And it is found in most of the angiosperm. ये जो maximum angiosperm है उनमें ये 8 nucleated 7 celled structure present होता है. This is the typical angiospermic embryo sac. Next comes types of embryo sac. So on the basis of variations, 3 main types of embryo sac development have been recognized. 3 तरह की embryo sac development study की गई है. First one is monosporic type. Second is bisporic type, and third one is tetrasporic type that we will discuss one by one in detail. Firstly comes monosporic embryo sac. As the name indicates, this type of embryo sac is derived from single functional megaspore. इस तरह का जो embryo sac है वो बनता है single functional megaspore से, as in case of polygonum, जैसा कि polygonum के केस में होता है. So this single megaspore mother cell will give rise to the entire embryo sac. All nuclei are genetically identical. सभी के सभी जो nuclei हैं वो genetically identical होते हैं और ये further 2 तरह का होते हैं. Monosporic embryo sac, first is Polygonum type and second one is Oenothera type. The first type of monosporic embryo sac is Polygonum type. This was the first studied embryo sac development which was noticed in Polygonum. In this case, firstly, the megaspore mother cell undergoes meiotic division to form 4 haploid megaspores, which are arranged in linear fashion. जो linear arrangement में arranged होते हैं linear fashion में. Out of these 4 megaspores, the megaspore which is present towards the chalazal end remains functional. ये chalazal end वाला megaspore functional रहता है, and remaining 3 megaspores disintegrates. वो degenerate हो जाते हैं. Then after that, this functional megaspores nucleus undergoes mitotic division, जिससे 2 nuclei बनते हैं और center में एक vacuole appear होना शुरू होता है that pushes these nuclei towards opposite poles. ये दोनों opposite poles की तरफ move करते हैं and at these opposite poles they undergo a repeated mitotic division twice. तो 2 बार mitotic division होता है resulting in formation of 4 nuclei at each pole. And out of these 4 nuclei, 1 nuclei from each pole moves toward the center, जिससे कि 2 nuclei central cell में arrange हो जाते हैं. And after that, this process of nuclear division is followed by cytokinesis. और यहाँ पे respected poles पे 3 cells बनते हैं towards micropylar end. It gives rise to the egg apparatus, जो बना होता है egg और 2 synergids से. While at chalazal end, it gives rise to antipodal cells. And in the central cell there are 2 nuclei which are known as polar nuclei. So इस तरह के embryo sac की development को बोलते हैं polygonum type, and this is one of the common, and this is also known as normal embryo sac. So here this functional megaspore elongates and nuclear division is not followed immediately by cytokinesis. So जो nuclear division है immediately cytokinesis नहीं होता है इसमें, and large vacuole appears in the center. Due to the appearance of this vacuole, it pushes the daughter nuclei to 2 poles. जो ये 2 nuclei बने हैं ये दोनों opposite poles की तरफ push होते हैं due to appearance of this vacuole. Then daughter nuclei divides mitotically ट्वाइस जो डॉटर न्यूक्लियाई दोनों अपोजिट पोल की तरफ मूव किए हैं, ये 2 बार माइटोटिक डिवीजन शो करेंगे, रिजल्ट इन फॉर्मेशन ऑफ 4 न्यूक्लियाई एट ईच पोल। तो 4 न्यूक्लियाई एक पोल में बनते हैं। आफ्टर दिस सेलुलर ऑर्गेनाइजेशन टेक्स प्लेस, इसके बाद होती है साइटोकाइनेसिस दैट रिजल्ट्स इन द सेलुलर ऑर्गेनाइजेशन, जिसकी वजह से बनते हैं सेल्स। एट माइक्रोपाइलर एंड ऑर्गेनाइजेज इनटू एन एग apparatus। जो माइक्रोपाइलर एंड है वहाँ पे जो 3 सेल्स हैं, दे विल फॉर्म एग apparatus, जिसमें एक एग है और 2 synergids हैं। while at chalazal and the nuclei organized, और जो cells हैं they organized to form antipodal cells, and 4th remain in cytoplasm of the central cell. So जो 2 nuclei हैं, one from this chalazal and other from this micropylar end, that they move to the center, और इन्हें बोलते हैं polar nuclei। ये present होते हैं central cell में। So इस तरह का जो embryo sac है उसे कहते हैं Polygonum type, and this is one of the most common type of embryo sac, which is 8 nucleated 7-celled embryo sac, and this is one of the common embryo sac which is present in angiosperms. The second type of monosporic embryo sac development is Oenothera type. In this case, this megaspore mother cell undergoes meiotic division, just similar to that of Polygonum type, results in formation of 4 haploid megaspores. Out of these 4 megaspores, either of micropylar end or of chalazal end remains functional, remaining 3 will disintegrate. इनमें से एक ही एंड पे, या तो माइक्रोपाइलर एंड पे या फिर chalazal एंड पे, मेगास्पोर फंक्शनल रहेगा, and remaining 3 megaspores will disintegrate. So this functional megaspores nucleus undergoes meiotic division, results in formation of 2 nuclei. Then जो ये 2 nuclei हैं ये उसी पोल में अरेंज रहते हैं जिसमें ये बनते हैं, ये मूव नहीं करते हैं अपोजिट पोल की तरफ। देन दे विल अंडरगो अगेन द माइटोटिक डिवीजन, और इसमें जो सेकंड माइटोसिस है वो एब्सेंट होता है, दैट रिजल्ट्स इन फॉर्मेशन ऑफ 4 सेल्स। आउट ऑफ दिस 4 सेल्स 3 रिमेन्स एट वन एंड, जो बनाते हैं एग apparatus, और एक ही मूव करता है टुवर्ड द सेंट्रल सेल, जिससे कि यूनिन्यूक्लिएट सेंट्रल सेल इज प्रेजेंट। इसमें यूनिन्यूक्लिएट सेंट्रल सेल बनता है और 3 बनाते हैं एग apparatus. So this type of embryo sac is 4-celled 4-nucleated embryo sac, 4-celled 4-nucleated embryo sac, और ये इस तरह के embryo sac है characteristic feature of family or Oenotheraceae. So this type of embryo sac is derived here from functional micropylar or chalazal megaspore of linear tetrad, या तो micropylar end का या फिर chalazal end का megaspore बनाता है इस तरह के embryo sac को। Both the nuclei after first meiotic division of the functional megaspore remains on the same pole. जिस पोल में ये न्यूक्लिया प्रेजेंट है वहीं पे ये रहते हैं, they remain on the same pole. Second division forms 4 nucleate structure, और second division से बनते हैं 4 nucleate structure. And of the 4 nuclei, 3 nuclei organizes into an egg apparatus, जो 3 है वो egg apparatus बनाते हैं, and the 4th one moves to the center of the center cell, जो central cell के center में move करता है 4th nuclei. In this way 4 nucleate and 4-celled embryo sac is formed, and this is Oenothera type of embryo sac, and it is a characteristic of family Oenotheraceae. ये है characteristic feature family Oenotheraceae का। अगर हम इसे briefly summarize form में लिखे Oenothera type of embryo sac, so it is an embryo sac with an egg apparatus and uninucleate central cell. तो इस तरह के embryo sac को बोलते हैं Oenothera type of embryo sac development. Next type of embryo sac development is Bisporic Embryo Sac. As the name indicates, this occurs due to failure of cytokinesis after meiosis 2. So there is formation of 2 cells which contain 2 nuclei each. Out of these 2 cells, one cell with 2 nucleus will survive and other will disintegrate, and this will result in formation of embryo sac. So bisporic development results from failure of cytokinesis after meiosis 2. जो meiosis 2 है उसके बाद जो cytokinesis का प्रोसेस है वो फेल हो जाता है, that results in formation of bisporic development. As a result, the megaspore tetrad is comprised of 2 cells. तो 2 सेल्स से मिल के बनता है मेगास्पोर टेट्राड, बट इसमें सेल साइटोकाइनेसिस नहीं होते। देयर आर 4 न्यूक्लियाई इन 2 cells, 2 in each cell, so each containing 2 megaspore nuclei. तो हर एक सेल में 2 megaspore nuclei that we can see in this diagram. The embryo sac develops from one of these diodes. डायाड्स, जो ये एम्ब्रियो सैक है ये बनता है इन दोनों में से किसी एक डायाड से. डायाड मीन्स हैविंग 2 nuclei cell with 2 nucleus by 2 rounds of meiotic division. This cell will undergo 2 rounds of meiotic division that results in formation of 8 nuclei. This results in formation of 8 nuclei and 2 central polar nuclei. So the embryo sac develops from one of these dyad cells by 2 rounds of meiotic division and the outer dyad cell is suppressed. और जो outer dyad cell है वो suppressed हो जाता है, that gets disintegrated. और mostly more commonly जो micropylar dyad है वो बनाता है embryo sac. 2 megaspore nuclei undergoes meiotic division to form 8 nuclei. 8 nuclei बनते हैं by repeated meiotic division. 2 meiotic divisions होती हैं इसके बाद, that results in formation of 8 nuclei. The nuclei in such embryo sacs are genetically different होते हैं, 4 of one type and 4 of other type, as they are derived from 2 meiotic products. तो ये 50% similar होते हैं. This bisporic embryo sac are of further following 2 types. 2 तरह के होते हैं ये bisporic embryo sac that we'll discuss. Firstly, we have Allium type. In this type of embryo sac, here the embryo sac is derived from chalazal dyad cell. तो इसमें जो meiosis के बाद 4 nuclei बनते हैं in 2 cells. So out of these 2 cells, जो chalazal end वाला megaspore है that contains 2 nuclei after meiosis 2, तो ये वाला cell survive करता है and it will give rise to the embryo sac. So the second meiotic division in the chalazal dyad is not followed by a cell wall formation. So second जो meiotic division होता है, it is not immediately followed by cytokinesis. इसमें cell wall formation नहीं होता है. The 2 megaspore nuclei undergoes 2 mitotic divisions. अब जो ये 2 nuclei वाला megaspore cell है, it will undergo further 2 mitotic division that results in formation of 8 nuclei. So that results in formation of 8 nuclei like this. After meiosis 2, 8 nuclei बन जाएंगे. These 8 nuclei organizes themselves into 3 groups similar to the Polygonum type of embryo sac. जैसा हमने Polygonum type of embryo sac में पढ़ा, वैसे ही ये 3 groups में अरेंज कर देगा. 3 plus 3 plus 2, that is 3 egg apparatus, 3 forms antipodals and 2 the polar nuclei are in the central cell. Example is Allium. So Allium में इस तरह के embryo sac development होता है. This is Bisporic embryo sac. Sometimes 2 chalazal nuclei fail to undergo second mitosis. As a result, the mature female gametophyte is 6 nucleate. So कई बार जो 2 chalazal end के nuclei हैं उनमें second mitotic division नहीं होती है, जिसके result से 6 nucleate का एक female gametophyte और embryo sac बनता है, as in case of Cypripedium कॉर्डिजेरम. तो एक प्लांट है ये साइप्रिपीडियम कॉर्डिजेरम, इसमें 6 न्यूक्लिएटेड एम्ब्रियो सैक बनता है. दिस इज एन एक्सेप्शनल केस. Second type of bisporic embryo sac is Endymion type. In this case, it is just similar to that of the Allium type, but in this case, embryo sac is derived from micropylar dyad. जबकि जो Allium type है उसमें embryo sac बनता है chalazal dyad से. In the Endymion type, जो embryo sac है develops from the micropylar dyad. This is the only difference. All other divisions are similar to Allium type. और सारे के सारे जो divisions है वो Allium type की तरह ही हैं. The examples of plants which contain this type of embryo sac are Endymion and Scilla. So these plants contain embryo sac development like of Endymion type. The next type of embryo sac development is Tetrasporic embryo sac. In this type of embryo sac development, after meiosis 2, the nuclear division is not followed by cytokinesis, that is division of cytoplasm, which results in formation of 4 nuclei present in a single cell. और इसमें जो meiosis 2 के बाद cytokinesis का failure होता है, it means cell wall formation नहीं होता, that results in formation of 4 nuclei. And all the 4 nuclei will participate in the formation of embryo sac. और इस तरह की embryo sac की development को बोलते हैं Tetrasporic embryo sac. So none of the meiotic divisions of megaspore mother cell during megasporogenesis is accompanied by any wall formation. So इसमें कोई भी wall formation नहीं होता है. As a result, all the 4 haploid nuclei remains in common cytoplasm, and that result in a structure which is known as cynomegaspore. That is just similar to that of cynocyte. It is a condition which contains multiple nuclei within a single cell. तो उसे कहते हैं cynomegaspore, the spore which contain more than one nuclei. So depending upon the arrangement of these 4 nuclei, the cynomegaspore, 3 main types of arrangements have been found in Tetrasporic Embryo Sac. 3 तरह की arrangement है, that is 1 plus 1 plus 1 plus 1 arrangement. Then second one is 1 plus 3 arrangement, and next last one is the 2 plus 2 arrangement. So on the basis of this arrangement, total 3 ग्रुप्स में हम डिवाइड कर सकते हैं जो ये मेगास्पोर के न्यूक्लियाई हैं उनके अरेंजमेंट के बेसिस पे. So these are 1 plus 1 plus 1 arrangement that is found in case of Pepromia, Penia, Plumbago. Then next arrangement is 1 plus 3 arrangement which is found in Drusa, Fritillaria, Plumbagella. And the last one is 2 plus 2 arrangement which is found in case of Adoxa. That we will understand one by one in detail. The nuclei in such embryo sac are more heterogeneous. It means they are not genetically similar. So they are genetically different. So they are further off 7 different types that we'll discuss one by one. So the first one is Pepromia type. In this case, in this Pepromia type of embryo sac development, after meiosis 2, within a single megaspore it contains 4 nuclei, और इसमें जो arrangement होती है that is 1 plus 1 plus 1 plus 1 arrangement, and these 4 nuclei are present at the 4 corners that we can see in this pic. And after that it will undergo 2 successive mitotic division. इसमें 2 successive mitotic divisions होते हैं, और जो nuclei हैं they are present at the corners. That results in the formation of a 16 nucleated structure जो कि 4 के ग्रुप्स में अरेंज होते हैं. And out of these 4 groups at the 4 corners, 2 nuclei they will move towards the center. 2 nuclei हर corner से center की तरफ move करेंगे. That results in formation of 8n secondary nucleus, जिससे कि 8n secondary nucleus बनता है. Then the remaining 2 nuclei which are present at the micropylar end, they will form egg apparatus. ये बनाते हैं egg apparatus. Out of these 2 cells, one is egg and one is the synergid, and the rest of the 6 cells they will form the peripheral cells. So इस तरह की जो embryo sac development है that is known as Pepromia type. So 4 haploid nuclei of the cynomegaspore, that is 1 plus 1 plus 1 arrangement, undergo 2 successive mitotic divisions. So 2 successive mitotic divisions होती हैं, that results in formation of 16 nuclei which are present in at a group of 4 at 4 corners. 4 के ग्रुप में 4 corners present है of the developing embryo sac. The 2 nuclei from each corner moves to the center. So 2 nuclei हर corner से out of 4 nuclei 2 nuclei move करेंगे center की तरफ, and they will fuse to form 8n secondary nucleus. जो secondary nucleus बनेगा that will be 8n. Remaining 2 nuclei at the micropylar end will form an egg apparatus which consists of an egg and synergid, while the remaining 6 nuclei are organized and give rise to the peripheral cells. So this type of development is found in case of Pepromia type. Next type is Penia type. This is just similar to that of Pepromia type. There is one difference. So the cynomegaspore, here again the arrangement is 1 plus 1 plus 1 plus 1 arrangement, and the nuclei are present at the 4 corners. They will undergo 2 successive mitotic division. That results in formation of 16 nucleated structure like this, और जो ये 16 nuclei they are present in a group of 4 at 4 corners, and out of these 4 groups, one single nucleus will move toward the center, and that forms the 4n type of secondary nucleus. And remaining cells, they are 3 3 in a group of 3 3. They are arranged at the four corners, and the micropylar end form the agapparatus. जो micropylar end वाला group है 3 cells का, that will form agapparatus. Out of these 3 cells, 1 is egg and 2 are synergids. So this type of development is found in case of Penia. So 4 haploid nuclei of the megaspore, that is arrangement of 1 plus 1 plus 1 arrangement, will undergo 2 successive mitotic division and results in formation of 16 nuclei arranged in groups of 4 at 4 corners, that we have studied already in Pepromia. So वैसे ही इसमें होगा, but the difference is 1 nucleus from each corner. Here only single nucleus from each corner will move toward the center and will give rise to the formation of polar nuclei, जो कि 4n होगा. So this is the difference, and remaining 3 nuclei at the micropylar end form the agapparatus, और ये बनाता है एक agapparatus, and the remaining nuclei are organized at 3 corners in the groups of 3 each. So this structure is the 16 nucleated 13 celled structure that we can see here. जो Penia है, that is 16 nucleated, 16 nuclei बने हैं, और 13 celled structure, 3 cells at 4 corners, that is 14, and 4 at the center. At the central cell, that is a single cell, so it is 13. While in case of Pepromia, it was 16 nucleated, 9 celled structure. Here 2 cells are the part of agapparatus, 1 is the central cell, and the remaining 6 are the peripheral cells. So this Pepromia is 16 nucleated, 9 celled structure. The next type of embryo sac development is Drusa type. In this case, in Drusa type of embryo sac, here a single nuclei, this is 1 plus 3 arrangement of megaspore, जिसमें जो 4 nuclei हैं उनमें arrangement है 1 plus 3 की. 1 is at one corner, 3 at the other corner. They will undergo 2 mitotic division. Successive mitotic division results in formation of 16 nuclei. Out of these 16, 4 are at one corner and 12 are at other corner. And out of these 2 groups, one nuclei will move toward the center. One nucleus will move toward the center and give rise to 2 polar nuclei to form the secondary nucleus. And remaining cells at micropylar end, there are 3 cells which form agapparatus, and remaining rest of the 11 cells are present at the chalazal end. So 4 haploid nuclei of the megaspore gets arranged in 2 groups, that is 1 plus 3, that we can see in this image. They further undergo 2 successive mitotic division and results in formation of 16 nuclei arranged in 2 groups of 4 plus 12, that we can see here, 4 plus 12 arrangement. 4 are present on micropylar end and 16 and 12 are present on the chalazal end. That results in formation of total 16 nuclei. 1 nucleus from micropylar end and 1 nucleus from chalazal end will move to the center and forms polar nuclei. So that will be the 2n after fusion. So 3 micropylar nuclei are organized to form an agapparatus. जो micropylar end वाले ग्रुप्स हैं, वो 3 सेल का ग्रुप है, वो बनाएगा agapparatus, जिसमें 2 होंगे synergids और 1 होगा egg, and 11 chalazal nuclei form the antipodals, और ये बनाते हैं antipodals. So this embryo sac consists of 16 nucleated, 15 celled structure. So this is this type of embryo sac is found in Drusa type. Next comes Fritillaria type. In Fritillaria type of embryo sac development, here again there is arrangement of 1 plus 3. तो 2 groups में present होते हैं nuclei of the megaspore, that is 1 plus 3 arrangement, and जो chalazal end वाले 3 nuclei हैं, they will fuse to form a single triploid nucleus. Then after that they will undergo 2 successive mitotic division, which results in formation of 4 haploid nuclei at one corner and 4 triploid nuclei at other corner. So that we'll understand. So 4 haploid nuclei of the megaspore gets arranged in 2 groups, that is 1 plus 3. तो इसमें 1 plus 3 के arrangement है Fritillaria, that we can see in this image. The 3 nuclei sent on the chalazal end will fuse to form triploid nucleus. ये बनाते हैं एक triploid nucleus, that we can see here. This is the triploid nucleus. Then they will undergo 2 successive mitotic division to form 4 haploid nuclei, जिससे 4 haploid nuclei बनते हैं at micropylar end, and 4 triploid nuclei towards the chalazal, और 4 बनते हैं triploid nuclei at the chalazal end, and this embryo sac is 8 nucleated embryo sac. One nucleus from each side moves to the center. हर एक न्यूक्लियस दोनों पोल से मूव करेगा टुवर्ड सेंटर. वन इज ट्रिप्लॉइड न्यूक्लियस, अदर इज हैप्लॉइड न्यूक्लियस. दैट रिजल्ट्स इन फॉर्मेशन ऑफ टेट्राप्लॉइड secondary nucleus, or central cell contain the tetraploid nucleus. So this is tetraploid secondary nucleus. Remaining 3 nuclei at the micropylar end will form an egg apparatus. ये बनाते हैं एक egg apparatus which consists of 1 egg and 2 synergids and the 3 chalazal triploid nuclei. और जो chalazal end पे 3 nuclei हैं, they will form the antipodals. So ये जो antipodals हैं, they are triploid. So this embryo sac is 8 nucleated 7 celled structure. So this is 8 nucleated 7 cell structure. So 3 are 3 cells are of an egg apparatus and 3 triploid antipodals and 1 central cell, so 7 cell structure. So this type of embryo sac is found in case of Fritillaria type. The next type of embryo sac is Plumbagella type. Here in this case, in Plumbagella type, there is again arrangement of these megaspore nuclei in 1+3 groups. One is at one corner, one nucleus is at one corner, and other 3 nuclei will fuse to form a triploid nucleus, and it is followed by a single mitotic division. इसमें एक ही mitotic division होगा, इसमें 2 mitotic division नहीं होंगे, जिसके रिजल्ट से एक एंड पे 2 हैप्लॉइड न्यूक्लियाई बनेंगे और दूसरे एंड पे 2 ट्रिप्लॉइड न्यूक्लियाई बनेंगे, एंड दे विल फॉर्म एन एम्ब्रियो सैक फ्रॉम these 2 ग्रुप्स. एक जो ट्रिप्लॉइड न्यूक्लियस है चैलेजल एंड से सेंटर में मूव करेगा और दूसरे एंड से एक हैप्लॉइड न्यूक्लियस मूव करेगा सेंटर में. दैट विल रिजल्ट इन फॉर्मेशन ऑफ टेट्राप्लॉइड सेकेंडरी न्यूक्लियस, एंड देयर इज ओनली 1 cell at the micropylar end that will form an egg, and at other end a single cell will represent the antipodal, which is triploid. So this embryo sac is 4 celled. This embryo sac is 4 nucleated. This embryo sac is 4 nucleated 3 celled structure. So this is very important. So this embryo sac is 4 nucleated 3 celled structure. So this type of embryo sac is found in case of Plumbagella. Here 4 haploid nuclei of the megaspore gets arranged in 2 groups, that is 1+3, similar to Fritillaria type. 3 nuclei are present on the chalazal and fuse again to form triploid nucleus that we have already studied in case of Fritillaria type of embryo sac. They undergo single mitotic division. इसमें repetitive 2 successive mitotic division नहीं होते हैं, single mitotic division होता है, and form 2 haploid nuclei towards the micropylar end. 2 haploid nuclei बनते हैं micropylar end में, और 2 triploid nuclei they are formed at the chalazal end. So this is a 4 nucleated embryo sac, and this is again a 4 nucleated 3 celled embryo sac. One nucleus moves to the center from one side of the tetraploid, from each side, that is one from micropylar end, that is haploid nucleus, and one from the chalazal end, that is triploid nucleus. That will give rise to a tetraploid nucleus, secondary nucleus. Remaining 1 nucleus at the micropylar end is form an egg. जो micropylar end वाला एक nucleus है वो बना देगा egg, and 1 chalazal and triploid nucleus will form an antipodal. So this type of embryo sac contains single triploid antipodal and single haploid egg. So this is a 4 nucleated 3 celled embryo sac which is found in case of Plumbagella type. Next type of embryo sac is Plumbago type. In Plumbago type of embryo sac, here again this is the arrangement of 1+1+1 arrangement of megaspore nuclei जो कि 4 कॉर्नर में प्रेजेंट होते हैं. दे विल अंडरगो अ सिंगल माइटोटिक डिवीजन. एक सिंगल माइटोटिक डिवीजन होगा जिसके रिजल्ट से बनते हैं 8 nuclei which are arranged at 4 corners in pairs में प्रेजेंट होते हैं ये. एंड आफ्टर दैट a single nucleus will move toward the center from each corner. That will give rise to a 4n type of secondary nucleus. 4 polar nuclei होंगे central cell में, and remaining 1 cell will at 4 corners. At micropylar end, a single cell will form egg, और बाकी rest of the 3 cells they are present at the 3 corners. So here 4 haploid nuclei of the megaspore they will undergo a 1 mitotic division. इसमें एक ही mitotic division होता है, results in formation of 8 nuclei. 8 nuclei बनते हैं arranged in 4 pairs at 4 corners that we can see in this image of the developing embryo sac. One nucleus from each corner will move towards the center and get organized into a 4 polar nuclei. और यहाँ पे 4 polar nuclei बनेंगे central cell में. Remaining one nucleus at micropylar end will form an egg apparatus or egg, a single egg. Remaining 3 nuclei are organized as peripheral cells on the 3 corners. और जो remaining 3 cells हैं वो present होंगे peripheral 3 corners में. So this type of embryo sac is 8 nucleated 5-celled embryo sac, which is found in case of Plumbago. It contains a single haploid egg at one corner and remaining 3 peripheral cells, which are again haploid. Then comes the last type of embryo sac development, that is Adoxa type. In Adoxa type of embryo sac development, here again it will undergo only one mitotic division. So here the arrangement of this megaspore nuclei is 2 plus 2. That is the third group, 2 plus 2 arrangement. They will undergo a single mitotic division, which results in formation of 8 nuclei. And out of these 8 nuclei at 2 corners, one nucleus will move towards the center and it will give rise to a 2 polar nuclei in the center cell, and 3 cells at the micropylar end will give rise to an egg apparatus, which consists of a single egg and 2 synergids, and remaining 3 cells at the chalazal end will form the antipodals. So 4 haploid nuclei of the megaspore gets arranged in pairs on 2 corners, and this arrangement is 2 plus 2, 2 at micropylar end, 2 at the chalazal end. They further undergo one mitotic division. एक mitotic division होता है इसमें, that will results in formation of 8 nuclei. They are arranged in groups of 4 on 2 corners of the developing embryo sac. One nucleus from each corner moves to the center. एक nucleus दोनों end से move करेगा towards center and get organized into 2 polar nuclei, और 2 polar nuclei arranged होते हैं in the central cell that we can see in this image. Remaining 3 nuclei at micropylar end forms an egg apparatus. जो micropylar end वाले 3 nuclei है, they will form an egg apparatus consisting of an egg and 2 synergids, and remaining 3 nuclei at chalazal end are organized into an antipodal, और वो बना देंगे antipodal. And this type of embryo sac is 8 nucleated and 7 celled structure. So these were the types of embryo sac development which is found in case of Tetrasporic embryo sac development. So ये examples थे कुछ जो हमने पढ़े. The next comes structure of a mature embryo sac, and that we have already discussed in our first slide. The mature embryo sac is most of the angiosperm is ellipsoid, that is the egg-shaped structure with tapering ends, and it is 8 nucleated 7-celled structure consisting of 1 egg, 2 synergids, antipodal cells and antipodal cells, and 1 central cell. So 7-celled structure है, 8 nucleated structure है. The wall of the mature embryo sac is multi-layered and pectocellulosic in its chemical constitution. The innermost layer is derived from the wall of functional megaspore. जो inner wall है वो बनी होती है functional megaspore से, और जो outer wall है that is composed of the crushed cell of nucellus, वो बनी होती है nucellus के crushed cells. Different components of the mature embryo sac can be studied under the following categories, like that contains egg apparatus, then antipodals and central cell. So इन headings के अंदर हम एक mature जो embryo sac है उसको study कर सकते हैं. Then the first comes egg apparatus. An egg apparatus consisting of 1 functional egg, nucleus and 2 synergids, and is located towards the micropylar end of the embryo sac. और जो egg है that is easily differentiable because this egg cell is larger in size as compared to the synergids. So firstly, egg. It is located towards the micropylar end of embryo sac and surrounded by 2 synergids on the lateral sides. और lateral side से दोनों तरफ से जो synergids हैं वो इसे surround करते हैं egg को. And it is larger than synergids, so it is easily differentiable from the synergids. Cell wall of the egg is thick towards micropylar end. जो egg की cell wall है वो thick होती है towards micropylar end, while it thins down towards the chalazal side, और ये chalazal side पे thin होती है. That shows differentiation. एग सेल इज विदाउट एनी सेल वॉल टुवर्ड्स द chalazal साइड. सो chalazal साइड पे कोई भी सेल वॉल प्रेजेंट नहीं होती है एंड इज सराउंडेड ओनली बाय प्लाज्मा मेम्ब्रेन. और chalazal साइड पे इस पे सिर्फ प्लाज्मा मेम्ब्रेन प्रेजेंट होती है. A thin and irregular wall is present towards the chalazal side in capsula. Capsula के केस में एक thin और irregular wall present होती है chalazal side पे. Otherwise, most of the cases में cell wall is absent at chalazal side of the egg. So polarity starts appearing in the egg during its development. So egg भी polarity show करता है, means differentiation in its two ends, that is micropylar end and chalazal end. The next, egg becomes highly vacuolated towards micropylar end. Micropylar end पे egg vacuolated हो जाता है. Then the chalazal side of egg contain dense cytoplasm. While जो chalazal side है वहाँ पे dense cytoplasm present होता है, single nucleus present होता है, and most of the cell organelles are present at the chalazal end. But there are some exceptions. Like, however, nucleus is present in the center in case of epidendrum एंड जियामेज. इनके केस में जो न्यूक्लियस है वो सेंटर में प्रेजेंट होता है, वेयर साइटोप्लाज्म इज इवनली डिस्ट्रीब्यूटेड. और साइटोप्लाज्म की कोई रिस्ट्रिक्टेड डिस्ट्रीब्यूशन नहीं है जैसे नॉर्मली होती है. इन प्लांट्स में इट इज इवनली डिस्ट्रीब्यूटेड. द सेल ऑर्गेनेल इज रिपोर्टेड टू अकर द एग. इन द एग आर क्लोरोप्लास्ट, माइटोकॉन्ड्रिया विद फ्यू क्रिस्टे, एंडोप्लाज्मिक रेटिकुलम एंड डिक्ट्यूजोम्स. तो सारी की सारी सेल ऑर्गेनेल्स भी प्रेजेंट होती हैं एग में. एग डेवलप्स फिंगर लाइक प्रोजेक्शंस. In some exceptional cases like we have Plumbago, here Synergids are absent. So जो egg है उसमें ही finger like projections appear होती हैं जैसे कि filiform apparatus होते हैं Synergids में, but Plumbago lacks Synergid cell. So that's why this egg cell develops finger like projections which are equivalent to filiform apparatus. So these projections are similar to filiform apparatus of the Synergids and appear to have taken over the role of Synergid in addition to its normal gametic function. So egg ने यहाँ पे Synergids का function भी किया है साथ में. So it takes the function of Synergids also in addition to gametic function. Next comes Synergids. So the Synergids are also known as helper cell. इन्हें helper cell भी कहते हैं because ये guide करते हैं pollen tube को towards egg, जिससे कि एक मेल गैमेट फ्यूज करता है egg के साथ, and they also provide an area for the discharge of pollen tube material. So these cells are pyriform. It means they are pyriform. Pyriform means pear shape or elongated cell present towards the micropylar end on the two sides of egg. तो egg को surround करते हैं दोनों lateral side से. Their tips are prolonged into hook-like or beak-like structure. इनके tips होते हैं beak-like और hook-like. In case of Gossypium, cotton, Torrania or Ziamase, most of the angiosperm cell wall incompletely surrounds the Synergids. जो सेल वॉल है वो इनकम्प्लीटली सराउंड करता है Synergids को, सिमिलर टू द एग. जैसे एग में chalazal एंड पे प्रेजेंट नहीं था, बट देयर इज सम डिफरेंशिएशन फ्रॉम एग. हियर इन दिस सेल देयर केसेस वॉल इज प्रेजेंट ओनली ऑन द माइक्रोपाइलर एंड व्हाइल द chalazal साइड इज नेक्ड. सो इसमें जो वॉल है वो सिर्फ माइक्रोपाइलर साइड पर प्रेजेंट है और chalazal साइड होती है नेक्ड. chalazal साइड इज एनवेल्प्ड बाय टू प्लाज्मा मेम्ब्रेन. जो Synergids हैं उनमें chalazal साइड पे दो प्लाज्मा मेम्ब्रेन होती है. वन ऑफ द Synergid, एक होती है Synergids की अपनी और दूसरी होती है सेंट्रल सेल की. सो ऑन chalazal साइड इसमें दो वॉल्स होती हैं, दो लेयर्स होती हैं प्लाज्मा मेम्ब्रेन की. सॉरी, वॉल्स नहीं, दो लेयर्स होती हैं. वन प्लाज्मा मेम्ब्रेन इसकी अपनी है एंड दूसरी लेयर है सेंट्रल सेल की. हाउएवर cell wall is present all over the Synergid in exceptional cases like we have Aquilegia and Capsula. So Aquilegia or Capsula में cell wall present होता है. This is an exceptional case. The next most prominent feature is the presence of filiform apparatus. So जो भी Synergids हैं उनमें ये filiform apparatus present होता है. This is the finger-like projection जो कि deep seated होता है in the cytoplasm और इसके बहुत important roles हैं. It consisting of finger-like wall projections towards the micropylar side of the Synergids and is made up of cellulosic microfibril network. और ये बना होता है cellulosic microfibril network से. The projections of the filiform apparatus extend deep into the cytoplasm और ये deep seated होते हैं in the cytoplasm. Synergids are vacuolated towards the chalazal side. Chalazal side में vacuolated होते हैं and have dense cytoplasm with the nucleus and other organelles towards the micropylar side. और ये भी egg की तरह polarity show करते हैं. That is opposite to the egg. जो polarity है इनकी वो है opposite to an egg. जो सेलुलर ऑर्गेनेल्स हैं वो जो इसमें प्रेजेंट होती है, plastids, mitochondria, endoplasmic reticulum, dictyosomes and are abundant on the micropylar side just below the filiform apparatus. That shows जो synergids हैं, these cells are metabolically active. So this indicates that the synergids are metabolically active cells. They are short-lived cells of the embryo sac. One of the synergids degenerates even before the entry of pollen tube into the embryo sac. एक जो synergid है वो पहले disintegrate होता है even before the entry of pollen tube inside the embryo sac. The second persistent synergid also degenerate after some time. Synergids elongates enormously in some cases, like we have Quinquemelium chilensis. Quinquemelium chilens, that is the member of family Santalaceae and reaches up to a length of 1200 micrometer. So इस केस में जो synergids हैं वो enormously grow करते हैं. Next comes functions of synergids. So they play a very important role. They excrete chemotactically active substances and direct the pollen tube towards the embryo sac. और जो embryo sac, जो pollen tube है वो embryo sac की तरफ move करती है. So they excrete some chemicals which are chemotactic in nature. So the degenerating synergids form the site for the discharge of pollen tube, और ये एक space provide करता है pollen tube के material को discharge करने के लिए. So degenerating synergids form the site for the discharge of pollen tube contents. Then filiform apparatus of the synergids helps in absorption and transportation. Absorption and transportation of nutrients material from nucleus to embryo sac. तो nucleus से embryo sac पे जो material है, nutrient है, उसको transport करने में भी filiform apparatus help करता है. So these are the function of filiform and functions of synergids. Next comes antipodals. So these cells are present toward the chalazal end of the embryo sac. और मोस्टली नॉर्मल केसेस में ये 3 ही होते हैं, बट इट्स प्लॉइडी लेवल इज वेरिएबल, ये सिमिलर नहीं होते सभी तरह के एम्ब्रियो सैक में. सो दीज आर प्रेजेंट टुवर्ड्स द चैलजल साइड ऑफ द एम्ब्रियो सैक एंड आर हाईली वेरिएबल इन देयर शेप एंड प्लॉइडी लेवल. सो प्लॉइडी लेवल और शेप में ये वेरी करते हैं प्लांट्स टू प्लांट. दे मे डीजेनरेट बिफोर और सून आफ्टर फर्टिलाइजेशन इन द मेंबर ऑफ फैमिली कैरियोफाइलेसी, पोटेमोजिटोनेसी, सैलिकेसी एंड वर्मिनेसी. सो जो ये जो फैमिलीज हैं इनमें ये या तो फर्टिलाइजेशन से पहले या फिर soon after fertilization they will disintegrate, but in some cases they persist and they are involved in nutrition of the developing embryo. So ये किसी जो कुछ embryo sac में persistent होते हैं and they provide nourishment to the developing embryo. Usually 3 antipodals are present in the most of angiosperms, जो एक normal type of embryo sac है, but the number can be as 300. So ये जो antipodals का नंबर है वो 300 भी हो सकता है in case of some grasses, for example that is Sassa paniculata. So ये जो grass है इसमें जो antipodals है इसका नंबर हो सकता है 300 तक. 3 antipodals in Commelina, Crocus, Gladiolus, Iris and Ornithogallum are exceptionally large in size. तो ये जो plants हैं इनके जो antipodals हैं उनका size exceptionally large होता है. So these are some unique cases. Next, the antipodals have distinct wall which remain thick and fibrous towards the micropylar end. जो micropylar end की तरफ इनका position है वहाँ पे जो wall है वो distinct होता है, that is thick and fibrous. However, antipodal cells are without any cell wall in Brassicaceae, Melastomaceae, Orchidaceae, Polygonaceae and Sapotaceae. So ये कुछ families हैं जिनमें antipodal cells में कोई भी wall present नहीं होता है. Then additional division in ziamades may form many antipodals with incomplete wall. In ziamades जो antipodals हैं इसमें additional cell division होता है that results in formation of many antipodals with incomplete cell walls. Such antipodal cells forms multinucleate protoplast or syncytium. So that results in formation of syncytium. Sometimes the antipodal nuclei become polyploid. इसमें पॉलीप्लॉइडी भी दिख जाती है ड्यू टू पॉलीटिनी एंड एंडोपॉलीप्लॉइडी. सो ये कुछ रीजंस हैं, दैट इज पॉलीटिनी और एंडोपॉलीप्लॉइडी, जिसकी वजह से जो एंटीपोडल न्यूक्लिया दे बिकम्स पॉलीप्लॉइड हैविंग मल्टीपल सेट्स ऑफ क्रोमोसोम. सो एंटीपोडल सेल्स आर रिच इन प्रोटीन, lipids, enzymes and ascorbic acid, but they are deficient in RNA and polysaccharide. So RNA और polysaccharide में ये deficient होते हैं. The abundance of plastids, mitochondria, endoplasmic reticulum, then multicisternal ictiosomes indicates that these cells are metabolically active. और ये जो antipodals के cells हैं ये metabolically active होते हैं because they contain these cell organelles. Sometimes antipodal also develops haustoria. हाउस्टोरिया एक एब्जॉर्बिंग स्ट्रक्चर है जो एब्जॉर्प्शन में हेल्प करता है, ऑल्दो देयर क्लियर रोल इज़ नॉट नॉन। इसका जो क्लियर फंक्शन है वो क्लियरली नॉन नहीं है। द नेक्स्ट कम्स फंक्शंस ऑफ एंटीपोडल्स। इसके क्या क्या फंक्शंस होते हैं? सो फर्स्टली दे ट्रांसपोर्ट द न्यूट्रिएंट्स फ्रॉम न्यूसेलस टू एम्ब्रियो सैक, दिस इज वन फंक्शन। ये ट्रांसपोर्टेशन ऑफ न्यूट्रिएंट फ्रॉम न्यूसेलस टू एम्ब्रियो सैक में हेल्प करते हैं। Then they play a nutritive role, especially when they are persistent. जब ये persistent है means they will stay after fertilization, so they provide nutrition to the developing embryo. They may store proteins, lipids, and carbohydrates to be used by developing endosperm and embryo. Then they secrete some growth substances which regulates the development of adjacent endosperm inside the embryo sac. So these are some important functions which are performed by antipodals. Next comes central cell. So central cell is the largest cell among all the cells of embryo sac. It is an elongated and highly vacuolated cell which ultimately gives rise to endosperm after fertilization. So it has large number of blasters, mitochondria, endoplasmic reticulum, ribosomes, polysome and dictysome. सभी तरह की cell organisms present होती हैं. This is the central cell. Central cell. Then the cytoplasm is rich in protein, lipids and carbohydrates, although it can have variable number of nuclei. The most common number is 2. जो हमने अभी एम्ब्रियो सैक की डेवलपमेंट पढ़ी, जो ये नंबर है ये वेरी कर सकता है, बट मोस्ट जो नॉर्मल टाइप ऑफ एम्ब्रियो सैक है उसमें सिर्फ 2 ही न्यूक्लियाई होते हैं। 2 nuclei present in the central cell fuse either before double fertilization, ये fuse कर सकते हैं before fertilization to form diploid secondary nucleus और remain free as polar nuclei. तो ये fertilization से पहले fuse करके secondary nucleus भी बना सकते हैं, and they can stay in the form of 2 polar nuclei also. After triple fusion, the second male gamete will give rise to endosperm tissue. जब triple fusion होता है, first male gamete will fuses with the egg that will give rise to diploid zygote. जो दूसरा male gamete that fuses with this secondary nucleus और ये बनाता है triploid endosperm. Cell wall outgrowths similar to filiform apparatus have been reported in the central cell of Capsella. So जो Capsella है इसके central cell में filiform apparatus की तरह outgrowths देखी गई हैं. Then in addition to this, these filiform type of outgrowths are found in Euphorbia and Helianthus also. So ये कुछ examples हैं जिनमें central cell में filiform apparatus की तरह outgrowths देखी गई. Next comes embryo sac haustoria. Haustoria is an absorbing organ which helps in absorption of food or nutrition from the adjoining tissue. So this is basically an outgrowth that helps in absorption of food from the adjoining tissue. जो adjoining tissue से food और nutrients की absorption में help करता है उस structure को कहते हैं haustoria. And embryo sac और its component have been reported to become haustorial in some cases. तो जो embryo sac है और इसके कुछ components हैं, they become haustorial, they develop these outgrowths. And these outgrowths serving as an additional organ of absorbing nourishment. तो ये एक additional organ of absorption में help करते हैं. It becomes additional organ of nutrient absorption. In case of Santalum album, that is the sandalwood, chandan, the chalazal end of embryo sac is modified into extensive cecum. एक स्ट्रक्चर बनता है that is known as cecum, leaving the antipodals quite behind, जो कि जिससे कि जो antipodals हैं वो बहुत पीछे रह जाते हैं. Similarly, the micropylar end of embryo sac is modified into cecum in case of Comendra. तो Comendra में भी जो micropylar end है वहाँ पे embryo sac modify हो जाता है this structure which is known as cecum. In exocarpus, finger-like processes develops on the surface of embryo sac. तो वो भी नरिशमेंट को एब्जॉर्ब करने में हेल्प करते हैं. सो एक्सोकार्पस में एम्ब्रियो सैक के सरफेस पे कुछ फिंगर लाइक प्रोसेसेस डेवलप होते हैं. इन सम केसेस सिनर्जिड्स आर मॉडिफाइड इनटू हाउस्टोरिया. सिनर्जिड्स मॉडिफाई होते हैं, दे बिकम द एब्जॉर्बिंग ऑर्गन which are haustoria in the members of family Astraceae and Santalaceae. Then antipodal cells they are also modified into haustoria in Argemon, then Haplopepus and Quinchemelium. तो इन प्लांट्स में जो एंटीपोडल सेल्स हैं वो मॉडिफाई होते हैं दिस एब्जॉर्बिंग ऑर्गन दैट इज हाउस्टोरिया. नेक्स्ट कम न्यूट्रिशन ऑफ एम्ब्रियो सैक दैट वी हैव डिस्कस. बहुत सारे स्ट्रक्चर्स हैं जो प्रोवाइड करते हैं नरिशमेंट एम्ब्रियो सैक को. सो मेनली दिस इज कैरिड आउट बाय फ्यूनिकुलर वेस्कुलर सप्लाई. तो जो फ्यूनिकुलस से वेस्कुलर सप्लाई आती है एम्ब्रियो सैक में, दैट प्रोवाइड नरिशमेंट मोस्टली विच टर्मिनेट्स एट द चैलेजल एंड, जो कि टर्मिनेट होती है चैलेजल एंड ऑफ द ओव्यूल में. Beside this, the chalazial tissue, beside the chalazial tissue, इसके अलावा जो न्यूट्रिशन प्रोवाइड करते हैं that is nucellus and integumentary tapetum which is known as endothelium, which are the other source of nutrition for the embryo sac, जो कि कुछ और sources हैं embryo sac के nourishment के लिए. Then antipodals, जो कि persistent होते हैं, they also provide nourishment to the central cell, while the synergids provide nourishment to the egg before fertilization. Embryo sac or its component have also been reported to become haustorial in some cases, दो जो हम ऑलरेडी पिछली स्लाइड में डिस्कस कर चुके हैं. सो चैलेजल एंड ऑफ एम्ब्रियो सैक इज मॉडिफाइड इनटू केकम सीकम इन केस ऑफ सेंट्रलम एल्बम और सीकम एट द माइक्रोपाइलर एंड इन केस ऑफ कमेंड्रा अम्बिलेटा that we have discussed in previous slide. Haustoria structures are also known to form from antipodal, synergids and endosperm in different plant species. So all these structures serves as an additional organ of absorbing nourishment from the surrounding ovular tissue to bring about the nourishment of embryo sac. So this was all about for our today discussion about embryo sac and its type and the structure of mature embryo sac, then all the components of embryo sac and how it gets nourishment and what are the various nourishment providing tissues in embryo sac. Hope you will get some idea from this presentation. If you have any questions, queries, and any suggestions, you can give it in comment section. Thanks for watching. Have a great day.